Hello everybody, this will be a bit of a different video for YouTube. Since I've begun this BitChute channel, I've been posting a lot more on there due to the more egalitarian algorithm, and I've been growing pretty well on there. So I've stitched together several videos I've made for BitChute as a teaser for what you can get on my BitChute channel. The channel itself will be linked in the description below, so sit back and relax and enjoy this little sampling of videos you'll find on my BitChute channel itself. Hello and welcome to the next BitChute exclusive video. Today we'll discuss the phenomenon of a plant sport. A sport is a mutated branch of a plant that has different traits than its parent plant. In this episode, we will discuss the many uses of sports, some famous sports, and the phenomenon of witches' brooms, which is another type of sport. So hopefully you're a good sport about a video about sports. Not the sport kind, but the plant kind. Puns. How is a sport made? Sports are made in meristem tissue. Meristem tissue being the origin point of all new leaves, buds, and stems, and roots. Some meristems get mutations that have different effects on the plant itself, altering the branch. These mutations can be caused by a wide variety of processes. These include epigenetic mutations and genetic mutations. Epigenetic mutations are mutations that alter the DNA itself, but not the sequence, but the surrounding structures. This includes histone modification and DNA methylation. The DNA mutations include transposons, point mutations, and other basic mutations, such as tandem repeats, point error mutations, insertions, deletions, and base mismatches. Transposons are genes that switch places in the chromosome under certain conditions. These are also known as jumping genes because they move from location to location. Point mutations are alterations to one single nucleotide within a genome. An insertion is a mutation that is caused by a piece of DNA being inserted into a location where it's normally not found. Under certain conditions, this can cause a frameshift mutation, which can alter a protein produced by the DNA. Deletions are a mutation caused by a piece of DNA being removed from the sequence. A tandem repeat is a part of the DNA where multiple copies of the same DNA base pair appear adjacent to each other. This normally occurs in the non-coding regions of DNA. Sports have been generally used to breed many different types of cultivars. Examples include the Satsuma orange, which is a seedless sport of a common mandarin. Another example would be the self-pollinating almond. Sports have also been used to breed different variations of the same variety of rose. Here is an example. Alright, here's a normal flower color, a nice purpley color, and here's the sport. As you can see, it's a lot more pinkish. This is pretty cool, huh? Pink. Purple. Now let's cover some famous apples that have some famous sports. The first apple we'll discuss is called Delicious. Delicious is a very sweet and aromatic and complexly flavored apple that had many sports. This cultivar has many sports that have been made from this plant. The first one is called Starcane. The primordial strain is a red apple with yellow stripes. The common red Delicious we see today is a sport of that plant. This strain, however, lacks many of the flavor traits of its parent. Most of the sports of Delicious have been selected for the red color rather than superior flavor. The other plant on our hit list would be Cox's Orange Pippin. Cox's Orange Pippin is an apple with a rich, aromatic, nutty flavor. This cultivar has been used to breed a great many varieties of apple, and it has produced several interesting sports. The first is called Crimson Cox. Unlike the Cox Orange Pippin, which has orangey skin, this one has red skin. Then there's the Queen Cox Orange Pippin, another sport of Cox Orange Pippin, that has the properties of self-pollination. And finally, the Cherry Cox Apple. This is a sport of Cox's Orange Pippin that has the flavor of cherry. As you can see, even with minor mutations within branches, you can get a lot of variation to make new cultivars from. Now on to the final topic of this video, which is brooms. Witches' brooms are plants that have produced branches that have many stems that are short and tiny sticking out of it like a big ball of small tiny stems. This can be caused by a number of factors such as infection by fungi or Phytoplasma species, which is a wall-less single-celled organism with an unorganized nucleus. It can also be caused by a mite infestation, adverse environments, and genetic mutations, as well as dwarf mistletoe. Those caused by genetic mutations can be propagated or stable genetically, and those mutated branches can be propagated to make dwarf cultivars of conifers. One of my profs, Mike Gladstone, actually mentioned that many of these witches' brooms were collected by shotgun, where they just shot these things down with a shotgun. This concludes our video on sports and witches' brooms. See you again. Hello and welcome to another agri-history monologue. In this episode, we'll talk about breeding methods. First, you'll cover pollination of grain crops and some legumes. First, you'll cover crossbreeding. This is where you cross two types of plants together to create a new plant. This is the F1 generation. For instance, you may crossbreed a wheat plant with good yield and good flavor with a wheat plant that has high disease resistance and drought tolerance to get a plant with a combination of all these traits. Following that, for open pollinated varieties of grain, they do something called selfing. You see, the first set of seeds produced by the initial crossbreed will have a lot of different variation, different combinations of different traits showing up. You then select one plant to gather seeds from when that plant self-pollinates. When you find a plant that has the traits you want in the combination you want, you let that plant self-pollinate and collect seeds from that pollination result. There will still be variation within the progeny, but some will resemble the parent plant in physical traits. You will then select one of those plants that has the traits you want and collect seeds from a self-pollination of that plant. And as a result, the same effect will happen. There will be some genetic variants within there, but more of the plants will resemble the parent plant you selected. And as you repeat this process, the number of plants that resemble the parent will increase, and the variation will go down, until the traits get more or less fixed. This usually takes 7 rounds of self-pollination. During the interim as well, you can find other variations within the plant specimens, some of which are actually better than what you were looking for, and you may want to select seed from those as well, either for cultivar making or for further crossbreeding. The sky's the limit, and that's one of the reasons why I dislike the use of the double haploid method in tissue culture for the purpose of creating breeding lines. Even though you save a lot of time on breeding line creation, you also lose all those opportunities that the mutation rate and different variations within the seedlings provide you. Which brings us to the next part of open pollination, breeding styles. Another method would be backcrossing. Let's go back to that wheat crossbreed. Let's just say that none of the seedlings had a correct ratio of traits you wanted. Perhaps they had all the disease resistance and stress tolerance of one parent but limited yield from the other parent. You can select seedlings from that population that you like and crossbreed back into one of the parent cultivars to increase the amount of traits found in that population in the resulting progeny. And if you find seedlings you like from that population, you can just go through the same process I mentioned earlier. These methods are usually used for breeding different types of legume or self-pollinating grains, such as barley, oats, and wheat on the grain side of things, and soybean, dry bean, and chickpea on the legume side of things. Now onto F1 hybrids. F1 hybrids combine the vigor of a landrace with the uniformity of an open pollinated line. What they do is they collect plants of open pollinated lines and crossbreed them together to increase the yield of both. This works for plants that can be made to self-pollinate but naturally cross-pollinate. Examples include corn, many vegetables, canola, and sunflower. The advantage of this for the grower is that you get the uniformity of an open pollinated line and the vigor of a landrace. This creates high-yielding varieties of plant. The main problem with this process is that if you collect seeds from these F1 hybrids, the following generations will gradually lose their vigor because they're cross-pollinating with siblings. This causes a gradual decline in vigor, as well as wild variation within the following generations afterwards. For a seed company, this is a built-in patent. Like everything else in life, it's all about trade-offs. You give up something to get something. Each breeding practice has its pros and cons, and it's up to you as a farmer or grower to pick the varieties you want. Now on to some fruit breeding methods. The first method is open pollinated seedlings. This is the oldest form of fruit breeding. What would happen is that a plant they liked would have seeds collected from it, and these seeds would be planted. And then you select the seedling you want to grow and graft and propagate. Once again, you gotta graft or asexually propagate fruit plants for a simple reason that there's so much genetic variation within fruit trees and fruit plants that each seed has the potential to produce incredible amounts of variation, leading to a multitude of different varieties. This method of fruit breeding allows you to know the mother parent, but not the father parent. But it's the easiest method to use. The second method is controlled pollination. This is where you select two plants and you crossbreed them, using controlled methods to make sure only the varieties you want to be bred together will be bred together. The final method includes sports selection and simulclonal selection. Now sports selection we'll cover in another video, next week in fact, but as for simulclone selection, when you use non-meristematic materials for tissue culture, like leaves, roots, shoots, etc., you get a lot more mutations. So if in cell cultures of plant cell tissue, you get a lot of really cool and weird variations. These variations can then be isolated to produce new plants when you grow a tissue culture sample into a full plant. Right now there's a researcher at University of Guelph trying to use this method to create a mutated variation of Honeycrisp that's resistant to bitter pit. Well, that's about all. See you on the next one.